Now we're recording, so to further elaborate on your question, 3 of you can't come to lab today, so make sure you guys check your lab, pre-labs. I left notes saying, hey, make sure you finish this and then leave me a comment when you're done so I know to grade it. And you never left comments. And then some of you just never even did the thing I asked you to do. You have to do all the hypotheses before you can come. So, sorry, it's too late, you can't come. So you're gonna wanna get with a classmate, make sure you get some data from them so you can complete the lab. And then I'll see if I can come up with some other stuff, extra stuff you can do to get you some of your points back. Because you can't get full credit if you're not coming to the lab. So think about that. Get in touch with me. And you guys just make sure you're paying attention. Like if you look at that pre-lab, and the last question in the pre-lab said, hey, did you go down and answer all the hypotheses questions, right? And if you were to look, if you read the whole lab, like I always say you should before you come to lab, you'll see there's a whole set of questions you hadn't done yet. And also, like I said, make sure you check your emails often. And maybe I can help you with this, because when I leave a comment on your labs, you should get an email that said, hey, someone left a comment. And if it's not, that's not your fault. We need to figure out what's wrong with your email. And also, the flip side of that, that's why I need you to reply to your comments, because otherwise I have no idea of knowing that you've corrected it and you're ready for me to grade it. So you guys keep an eye on those things and let me know if I can help you. What else to say? Yeah. Before you turn anything, meet me online. Say, hey, before I hit the submit button, can you just look this over? You know, we can just meet online, I can look it over, and then I can just tell you, like, oh yeah, but don't forget this. I can verbally tell you. So really, you won't have any way of messing it up. I'm here to help you. Take advantage of it, please. And anyway, we start logging in here. Start doing attendance. So Friday, for example, after lecture on Friday, if you want, I'll be on office hours at least until noon. Why don't you just log on, come visit me online, share your screen with me, let me see your assignment, and then I can review that and I can tell you right then, and make sure you have it done right. Anyway, let's see. About 5 minutes. Is Aki here yet? No. Blount. Boggs is here and WVSU here. Bonner's here and WVSU here. Clay, right? Here. Clayton, not here. Dawson's not gonna make it. Ellis, not here yet. Paulo's not here. Green, I don't think is gonna make it. Harrison is here. Hinson is here. Grace is here. Esterson's here. King, right? King is here. Lewis is not here. Linda Q's not here yet. Morrison is here. Grace. Grace? Yeah. Yeah, 2 Graces. Sometimes I really confuse. And Ward is not here yet. Wilcox is not here. Wilder's not here. Parks is not here. Anybody I miss? I mean, I'm sure there's people online. I'll take a look there in a second. Tom's not here. The Bonner, Wilder, and Dawson are online. And if you're online, don't forget you have to send those attendance words so I know you were paying attention so I can mark you as present. I know some of you at least logged on for the past couple of meetings but didn't send the words. Granted, it's extra credit, so it's not gonna hurt your grades, but when I have to report attendance, that's where that comes in. Love this PowerPoint. Morning. Why is it backwards like this? Somebody screwed with the settings. All right, so Lewis, right? He was here. Wilcox is here. Ward is here. And then Ellis, right? All right, see if I can fix this PowerPoint since it's now time to start. What thing? I'm thinking, yeah. Thanks. Lewis is here, and then be issued. All right, bear with me while I fix this. I don't know why people mess with this stuff. All right, maybe that makes it. All right, guys. You know what? I don't want to waste any more of your time, so we'll just do it like this. It's a little odd, but it is what it is. Hopefully, if you're online, you can see this. You don't have an issue with screens. But we'll just do it like this. All right. So to remind you, we were just finishing up talking about proteins. Actually, let's back it up even more. This portion of chapter 2, we're talking about biological molecules. So we're done talking about carbohydrates. We're done talking about lipids. And we are finishing up talking about proteins. This particular slide, one of the things I want to remind you about proteins, it's very important about their shape, right? For all molecules, the shape is important. But like we were saying, very important, even more so for proteins. And this is where we left off, pointing out that protein shapes are sensitive to the environment. And actually, the first word for attendance, it's such a pain. I'll change things up a little bit. This time I'm going to point with the first word for attendance. So that's the first word for attendance. That little arrow points. Anyway, yes, so proteins, again, Their shape is very important, but their shape is also very sensitive to the environment. For example, a change in temperature might change the shape. And what I haven't taught you about temperature yet, I've taught you a little bit. Remember when I said something's hotter, for example, when I say the temperature is increasing, what I mean at the molecular level is that the molecules are moving faster. So we can expand on that a little bit, and I can tell you this. You can see these bonds, these little tiny dots represent hydrogen bonds, and if you remember, as you should remember because it'll be a test question, hydrogen bonds are the weakest of the bonds, and that's what we're looking at. So what happens if you add enough heat, enough temperature to something, those molecules start moving faster, and eventually they move fast enough, basically, to where they break their bonds. So again, when a protein gets too hot, that's what happens. You can break those bonds, and then obviously if it's break the bonds, if those things unravel, they change the shape, and therefore it doesn't work. And actually the term for that is called denatured. I'm not going to ask you that, otherwise I have it on the screen. But if a protein basically melts, it's called denaturing. So yeah, temperature is one thing that it can do it. pH is another example. So once again, look at this picture. We have, and it might be a little bit hard for you guys to see, but these little red things, if you remember, are oxygens, right? And the little white things are hydrogens, right there. Now, we know that oxygen end is slightly negative, remember, because it's a polar molecule. We know that hydrogen end is slightly positive. Now imagine if you change the pH, right? Let's say it was something, let's say you put that particular protein in something really acidic. What does that mean? What is there going to be a lot of then in that solution? So basically, remember, this is a definition you need to know, like what is an acid? If I say, oh, it's an acid, it has a lot of what ions? Which kind of ions? Good, hydrogen ions, right? So if you put a bunch of H pluses in here, anyway, if you put a bunch of H pluses in there, now remember, that thing is fully positive, right? So this isn't like some kind of hydrogen bond where that oxygen's slightly positive or negative, and that hydrogen's slightly positive. It's not, right? This is fully positive. So you can bet that fully positive hydrogen ion is going to disrupt those hydrogen bonds, therefore making it fall apart. Same thing if it was too basic, right? OH, there's a bunch of OH minus. Again, that's fully minus, right? That's a full negative charge. So that thing is really going to disrupt these hydrogen bonds, and therefore it'll rattle. Any questions about that? All right, that means we're done talking about proteins. So we're moving on to the fourth and final category, and that is nucleic acids. I'm going to go through this one kind of quick, relatively quickly, because I'm also going to go light on this portion for the exam. Because later in the semester, we're going to come back and talk about nucleic acids again, and we're going to look at them a little bit more deeply. So for now, consider this an introduction. There will be some questions, but again, this is basically an introduction to what you're going to learn later in the semester. So what do nucleic acids do? They store information. They provide instructions for building proteins. You do need to know that. And remember, in this exam, this multiple choice exam, I'll describe something, and you need to, based on that description, figure out what kind of molecule it is. And this is the only one that I've said where we store information, right? We can say that about proteins or carbohydrates or lipids. We also didn't say anything about them providing instructions for building proteins. So nucleic acids are the only thing where we would say that. And the reason I bring it up like that is because I want to remind you, for example, we said that carbohydrates store energy, right? But we also said fat stores energy, right? So for some of those other ones, the things they do kind of overlap, right? So this one's very specific, very unique. None of the other ones store information. None of the other ones provide instructions for building proteins. You definitely need to know this. The two types of nucleic acids are DNA and RNA. As far as I'm concerned, again, for this first exam, you can just know the letters, DNA, RNA. Later in the semester, when we start looking at them a little bit deeper, then you'll need to know the full name, deoxyribonucleic acid or ribonucleic acid. I might use the full name anyway. I don't know yet. When you think about it, that's almost like a dead giveaway. So if I was asking you questions, it's right there in the name, nucleic acid, right? So obviously, if we read that and say, oh yeah, that's obviously nucleic acid. That's the name. All right, any questions so far? Just to have more information about this coming up. All right. Next thing you need to know is what a gene is. So again, remember one of the purposes of nucleic acids is its instructions on how to build a protein. So that's where we have this term right here. Gene is a unit of inheritance that is encoded in the specific stretch of DNA that programs the amino acid sequence of a polypeptide. I really wish we could have gotten here yesterday, actually Monday. Because Monday, remember we talked about what a polypeptide is. So this is just the long-winded way of saying, let's see if I can draw here. Remember, and this is a good review for something you guys do need to know for the exam. The amino acids are what builds proteins, right? So you might have like a square amino acid, followed by a triangle amino acid, maybe by a circle, then another circle. I'm just making it up. It's not actually that easy. But basically, remember that's what a protein is. It's just a string of amino acids formed together to form a polypeptide. And then once it gets folded into its particular shape, then we consider it a protein. So what I'm saying then, with that in mind, that refresher in mind, a gene is just the thing that says, all right, first bring me this amino acid. Next thing you need is that amino acid. Next thing you need is this amino acid. Does that make sense? It's just instructions on which amino acids are going to be added to one another to make a polypeptide. The instructions are written in a chemical code that has to be translated. We go from the nucleic acid language to the protein language. The question mark next to this. I really don't think I'm going to ask you that on this first exam. We're going to have a whole chapter. Yep. Yeah, what chapter is that? It's chapter 10. The whole chapter that we're going to learn later is basically summed up on that one bullet point right there. We go from nucleic acid language to protein language. We learn all about that. There's names for each of the steps. There's certain ways that happens, and you can learn all about it. But for now, just know that. We go from nucleic acid language to the codes in the DNA that say, all right, we need this amino acid followed by this amino acid, so on and so forth. Any questions about what a gene is? The next slide is going to be a picture of a gene. This is from the old textbook, but I assume that the picture in the current textbook will look similar, right? So here's a long strand of DNA. Actually, you can't see the long strand, but imagine it's really long. We've just zoomed into this particular portion of it, and that part right there is a particular gene that provides instructions on how to build a protein, right? That gene, that information in the DNA is basically copied into something called RNA, and then finally the information in that RNA says, all right, bring me this amino acid, now bring me this amino acid, now bring me this amino acid, right? So that's instructions on how to build a protein. As far as this exam is concerned, that's all you really need to know. Gene provides instructions on how to build a protein. And again, later in the semester, then you're going to need to know, first we start with the DNA, then we go to RNA, then we go to protein. But for now, the gene is something that provides instructions on how to build a protein. Any questions about that? All right. Yeah, this is I mentioned this. This is from your textbook. Nucleic acids are the key macromolecule for the continuity of life. Again, that's instructions on how to build a protein. Or another way you could look at it, when you think about your whole genome, right? Your DNA is instructions on how to build you, right? Everything that you are, instructions on how to build you were in your DNA. We'll get into more detail later this semester, but long story short, all those proteins I keep talking about, the genes making, well, those proteins are the things that help build you. So those proteins are the things that make you who you are. The instructions on how to build those proteins are in your DNA. It is the key macromolecule of the continuity of life. What does that mean? That means it carries genetic information. Like I've already said, it carries instructions. Now that I'm looking at this slide, it's a little bit of a duplicate. I apologize. Same stuff, slightly different worded. So again, the two main types of nucleic acids, DNA and RNA. Any questions so far? All right, the next word for attendance, I'll just say it this time, life. Life is the next word for attendance. And remember, if you're online, you have to send it to get attendance to be counted as present. And then for everybody, again, these are extra credit. So point-wise, it'll do nothing but help you. All right, we'll talk a little bit more about this. Again, The DNA is the genetic material. It's found in all living organisms. I'm going to go past that pretty quickly because I'm going to ask you that later. And it's usually the RNA that's used directly used in protein synthesis. And again, I'll ask you that later. Yeah. See what I put myself a little note here. You'll learn more about this later. Oh yeah, independent work ideas. What else does RNA do? Oh yeah. So for independent work, because you don't need to know it, but you can look it up if you want. I'm telling you right now that RNA is involved in protein synthesis. But I'm also telling you, for your own knowledge, there's different types of RNA that actually do other things. And if you want to look into it further. It's a good reminder too. So one of your classmates asked this last Monday, I think. It's a good question, good time to remind you. Independent work is not extra credit. If you have to write these papers, you got all semester to write 100 points for the papers. It's not extra credit, it's mandatory. It's a full letter grade. If you need help with it, let me know. Here we go, back to this thing, right? Remember I told you a lot of it's going to be comparing and contrasting the different types of biological molecules, and I said one of the ways that we're going to do that is you have to know the polymer and the monomer. So here we go. The polymer of a nucleic acid is called a polynucleotide. Monomer, I can't talk today again. Monomer is just called a nucleotide. See, I love this. This one's easy, right? The words just make sense. It's we're talking about nucleic acid. So you have a nucleotide, that's the monomer. You have a bunch of them, it's a polynucleotide, nice and easy. Nothing tricky like the protein where, we put a bunch of amino acids together and you get a polypeptide. And that's not even technically a protein until it's folded into its correct shape, right? So it's nice and easy in my opinion. Now, remember, when we talked about the amino acids, I said you need to know the parts, right? Remember I was talking about that one central carbon that was bonded to a hydroxyl group and a side chain, right? And I said you had to know that shape, not because I was going to ask you to draw it, but I said I'm going to use that as a way to describe it, and then for that description, you should recognize it as an amino acid. Same thing here. So once again, in the exam, I'm going to describe this, and with this description, you should recognize that. Say, oh yeah, that's the description of a nucleotide. So again, to remind you and hopefully put you at ease for this 100 level multiple choice exam, you're not going to have a question where I say, tell me all the parts of a nucleotide, right? I'm not going to do that. I will say, here's these parts of a molecule, a 5-carbon sugar, a phosphate group, a nitrogen-containing base. What am I describing? Okay, from that description, you should recognize it as a nucleotide. And I put this part in bold. Sugar, phosphate, base. In my opinion, if you just remember those 3 things, that works. Yes, technically the carbon's a 5-carbon sugar. Yes, technically it's a phosphate group, not just a phosphate. Yes, technically it's a nitrogen-containing base. As far as I'm concerned, sugar, phosphate, base. 3 parts of a nucleotide, that's all you need to know. This third bullet point is worth pointing out, though. It's only the base that varies, right? So the 5-carbon sugar for all DNA, that 5-carbon sugar is exactly the same. For all DNA, all the phosphate groups are the same. But depending on the nucleotide, you might get a different base. So in a sense, you could almost think of the base as sort of like the side group of the amino acid. Remember, I said amino acid, they're all exactly the same other than that side group. That side group is sort of what defines the amino acid. Remember I told you there's 20 different amino acids. They're all exactly the same other than the side group. Now I'm telling you the same thing basically with these nucleotides. They're all the same. The other 2 parts of the nucleotide are the same. It's that third part, the base, that defines it. And that'll be much more important later in the semester. For this first exam, I'm going to probably go over again pretty light on that. Are there any questions about this slide? All right, here's another picture of a nucleotide. You can see the 5-carbon sugar, see the phosphate group, you can see the base. This is the same, a picture of the same thing. It's drawn a different way. So here you can see each little chemical bond. Here, it's just simplified, right? And once again, I'm not going to ask you any specific questions about this, right? I'm not going to give you this picture and say, without any labels and say, show me which one's the phosphate group, show me which one's the sugar, show me which part's the base. I'm not going to do that. Matter of fact, it'll probably be no pictures. don't remember. I have to double check. But if I did, again, I'm not going to ask you to name the parts. Any questions so far? Yeah, quiet today. Questions are good. I've been teaching this for years, so sometimes I know I'll probably go too fast because my brain, I'm like, oh yeah, this is just the 4 things. Duh. But you guys who maybe haven't seen this before, slow me down if I did. So anyway, like I said, it's the nitrogenous base that varies. So for DNA, there's one of 4 nitrogenous bases. So those are your choices when it comes to DNA. A, G, T, or C. So the same way amino acids, remember I told you there's 20 different varieties of amino acids that we can put together to build a protein? Well, DNA is 4 varieties of nucleotides. And that's the 4, A, G, T, and C. And again, for this exam, if anything, just knowing the letters will be fine. Because we're going to go into this later in the semester. Later in the semester, then you might need to know the full name. But probably not even. So yes, A, G, T, and C are the 4 bases for DNA. Any questions? Any questions about that before I show you a picture of it? All right, picture of it. There's your A, G, your T, and your C. Then there's a bunch of them put together, right? That's a double helix molecule of DNA. 2 DNA strands put together and wound up, which we'll talk about later. Another thing I'm not going to ask you to do is to recognize which one's A, G, T, or C. Right? You don't need to know that. You might notice that some of them, the A's and the G's, have 2 rings, right? And those T's and the C's only have 1 ring. That'll be important later in the semester, but for now, you don't need to know that. And even then, you don't need to memorize which is which. But the fact that those are 2 rings, these are single rings, that will be important later when we talk about how DNA fits together. Again, for now, all you need to know is A, G, T, and C are the 4 different bases for DNA. So, any questions about that picture? Right, that picture's not from your textbook, so if you wanted to study it and look at it later, you need to download it. All right, this is also not in your textbook, so I'm going to go through it pretty quickly. I did want to include this. As you might imagine, the chemical reactions that are bringing these nucleotides together, you should see a theme here, are called dehydration reactions. Right? So throughout this chapter, we keep talking about that. What is it when you bring 2 monosaccharides together? Dehydration reaction. What is it when you bring amino acids together? Dehydration reaction. What is it when you attach fatty acids to a glycerol molecule? Dehydration reaction. Right? So it's these dehydration reactions, in this particular case, we're bringing all these nucleotides and putting them together. That's what converts nucleotides to polynucleotides, right? Just putting a bunch of them back together. I'm going to put it next to this. I'm definitely not going to go into this much detail on this first exam, maybe later in the semester. But it is covalent bonds that are linking these polynucleotides together, and later you will need to know that. Later, not only do you need to know that they're covalent, but you need to know it's from the sugar of one nucleotide to the phosphate of the next. But again, for now, you don't need to know that. For now, you just need to know the nucleotides come together in dehydration reactions. But when the time comes, and when you do need to know that, it'll hopefully be easy to remember because there's something called a sugar-phosphate backbone. So when you look at pictures of DNA, when they're all linked up, all the nucleotides are linked up, we call that a sugar-phosphate backbone. So those are the parts that are bonded together. The bases, remember one of the other 3 parts, that part kind of hangs off like an appendage. Right? So it's not involved in those covalent bonds that are holding it together. And again, for this exam, not very important. It will be very important later in the semester. So any questions on that slide? Okay. Again, this is not your textbook, but I wanted to include it anyway. DNA is a double-stranded polynucleotide that is coiled around each other. And what I mean by that, and this again will be more important later in the semester, you have, when you see that picture of the double helix, you know, that twisty thing of DNA, what that actually is 2 strands of DNA that have come together and with hydrogen bonds, and again, that'll be later, and they've twisted. So next slide, I'll show you a good picture of it, but yeah. That will be important for you to know later, is the fact that when we're looking at DNA, we're actually looking at 2 strands of polynucleotides that have come together in a hydrogen bond. And another thing that you're gonna need to know later in the semester, not this one, not this exam, but they always pair up a certain way. A always goes with T, G always goes with C. And later, you're gonna really use that, but for now, this is just a preview. Here's a picture of what I'm talking about. That's actually two strands, right? These are two different strands. They've come together with these hydrogen bonds. Again, that A is hydrogen bonded to a T, C goes with G, so on and so forth. So they've come together and the whole thing just twists. Okay, so that's it for DNA. Any questions about that? We'll make a good time. So let's also talk about the similarities between DNA and RNA. And again, this will be important later in the semester. This is not in your textbook for this chapter, but it will be later, so I just wanted to go ahead and introduce you to it. So similarities. They're both polymers and nucleotides, right? So whether you're talking about DNA or RNA, they're all polynucleotides. The nucleotides all consist of sugars, phosphate, and bases, but there are some difference between the sugar and the bases, and we'll just talk about that later. But as far as what we need to know now, again, whether we're talking about DNA or RNA, those nucleotides, those building blocks, they're made up of the three parts: sugar, phosphate, base, right? That's the same. Any questions about that side? Okay. So those are the similarities. What are the differences between DNA and RNA? There's other ones, but these are the most basic. And again, not your textbook in this chapter. It will be later. This is a preview. RNA has a different sugar. So even though they both have the sugar, it's the 1/3 part of the nucleotide. RNA has a different type of sugar than DNA. Also, RNA does not have the T nucleotide, base. It has a U instead of a T. And again, that'll be important later. And then finally, generally speaking, RNA is single-stranded and DNA is double-stranded. There are some exceptions to that, depending on how you look at it. Sometimes a single strand of RNA will kind of fold up on itself, which makes it appear double-stranded, but that's stuff we'll worry about later. Any questions about this slide? All right, a picture of RNA, of an RNA nucleotide. So again, phosphate group's exactly the same. The sugar is different. As far as you guys are concerned, the sugar, it doesn't matter, right? It's chemically, they do the same thing as far as we're concerned with this 100 of course. And then again, that's the base. And in this particular instance, it's a U, because we're talking about RNA. Questions about that slide? Man, make a real good time. So really quickly, I will download the next PowerPoint. And then that situation, it'll show you where it is anyway. I'm sure you guys already know by now, but just in case, I'll show you where to find your PowerPoints. In theory. There we go. So have a walk in. So I guess the next word for attendance, sitting here waiting, is going to be PowerPoint. Use that one again. I'm trying to get our PowerPoint down. Oh, that brings me to a point. Make sure when you're doing, when you're watching those videos and they're asking you a question like, hey, what's your name and which course are you taking? Please do it. Like, make sure you put your full name and then the course, the actual course. Somebody from a different class said that they were in the 10103 class. They said they were in this class. And it just took me a few minutes. It took me too much time out of my day because I was an idiot. I thought, I don't remember him being in this class. And I was freaking out a little bit. Like, how do I not remember a student? Why do I not have their information? Turns out they were in 108. Anyway, Module 1, Chapter 1 and 2. Okay. So technically, this next chapter, Chapter 2, excuse me, let me get this down here. Yeah. Chapter 2, Chapter 3, Cell Structure and Function. All I want to do is basically introduce you to this and then actually really jump into it on Friday. Because I want you to be able to kind of marinate on what you've learned for chapter 2. Yeah, some of you are already looking at it. Good job. So one other thing to remind you. So this chapter that we're about to start is the last chapter that's going to be on the first exam. Chapter 1, chapter 2, chapter 3, what the first exam consists of. And it's downloaded. There we go. And once again, it's messed up, so I'll have to look a little bit on this side. Here we go. All right. Cell structure and function. Now, this used to be chapter 4 in the old textbook, and honestly, I used to skip it. So I'll just go ahead and let you know now. I'm going to go through this very quickly. It's basically, well, as the name indicates, you're going to learn about how the parts of the cell and how a cell works. However, in my opinion, this chapter is, I won't say useless, but it's not as useful because after this chapter, then you're really going to learn about how cells work, right? We're going to have a whole chapter on how cells reproduce. We're going to have a whole chapter on how cells get their energy. We're going to have a whole chapter on how DNA works in cells. So really, in my opinion, this is almost just like a preview of the more important stuff that you're going to learn later. And you are going to learn the more important stuff. So some of the stuff on here, in my opinion, is less important because you're not going to need it for the rest of the semester. So keep that in mind as we're going through this fairly quickly. And again, I'm going to go light on this one on the exam. Here we go. This is probably hard to read, but this is how the chapter's broken down. The first part is how cells are studied. And I'm really going to go through that quickly. Well, you'll see. Then the next part is going to be comparing prokaryotic and eukaryotic cells. After that, we're going to talk specifically about eukaryotic cells. Then we're going to talk about the cell membrane. Then we're going to talk about passive transport. Then we're going to talk about active transport. And just so you know, it's probably these 3 bullet points here. Oops. If I draw her. Those 3 bottom bullet points are the ones that you are going to need for next week's lab. It's going to be all about these 3 things. Cell membrane, passive transport, active transport. Anyway, let's jump into 3.1, how cells are studied. So the learning objectives of this particular section are, by the end, you should be able to describe the roles of cells in an organism, right? So what do cells do, basically. You should be able to compare and contrast light microscopy and electron microscopy. According to the book, those are the learning objectives here. And then finally, you should be able to summarize cell theory. And again, I teach out of the textbook, but I'm telling you right now, in my opinion, this is less important. So I'm going to go very light on it for the exams. For example, microscopes. There's different types. You've got light microscopes, dissecting microscopes, which are both different types of light microscopes. And then finally, electron microscopes. You guys aren't going to need that. We don't even use microscopes in this course. Of course, if you ever wanted to, they are back there. So if you ever, for some reason, want to do that during a lab, let me know. I can get some slides for you. But anyway, yeah, so those are different types of microscopes. I'm not going to ask you about that. I let myself know what to say. These are just the types from your textbook. They may be called something different depending on your source. Another great independent work idea, right? So I'm telling you that the book calls them this. The book says these are the 3 different types of microscopes. But if you want to look into it, find another source. And what are some other types of microscopes? And again, there are questions about this on the study guide, or the quiz, if you will, but not the exam. Anyway. You're definitely not going to ask you all the different parts. So you can see eyepieces versus the objectives, the stage, course focus. I'm not going to ask you any of that stuff. And again, if you are interested in that, I'll be glad to teach you. But I'm not going to teach all of you right now because that's a waste of your time. Cell theory, you do need to know this. Unified cell theory states that all living things are composed of 1 or more cells. Cell is the basic unit of life, and all new cells arise from existing cells. Yeah. You probably do need to know that. Actually, let me take that back. When you think about it, you do need to know that in a different context. We already talked about two things that you need to know from chapter 1 that are also up here. All living things are composed of cells, right? We said that's one of the properties of life. So you already need to know that for chapter 1. The cell is the most basic unit of life. You also need to know that as the property of life, right? So the only thing new here is the fact that all new cells arise from existing cells, which I think you all knew, right? Someone, I think, wrote a paper about what they used to think. Like if you had some rotten thing in a jar, that would produce maggots because they didn't know that maggots actually came from fly eggs that they couldn't see. But yeah, now we know, as I'm sure you all knew before I told you, every cell you see came from a cell prior to that. And then, of course, there's some people you can look into. Leeuwenhoek, Robert Hooke. I'm not going to ask you any questions about that. This is not a history of biology course, but you can look into them if you want. I thought I had some good documentaries. I'm going to look again. Have you guys seen the list of videos? Hopefully you have. If you click that link that says, I've got that document that says extra credit, that explains how extra credit works, but then there's another link that says, hey, here's a list of videos you can watch. So it's a good way to get extra credit, I think. Anyway, that's it for 3.1. We can jump into 3.2. We have time. 3.2, comparing prokaryotic and eukaryotic cells, right? You need to know the difference between prokaryotic and eukaryotic. Not that I expect anyone to know yet, but you know which one we are. Prokaryotes or eukaryotes? Does anybody know? Good, eukaryotes. So the learning objectives of this particular section are, you should be able to name examples of prokaryotic and eukaryotic organisms. We just did one, right? We are an example of a eukaryotic organism. We'll talk about some other examples. You should be able to compare and contrast prokaryotic cells versus eukaryotic cells and describe the relative size difference between them. Pretty simple stuff. And once again, for this 100 level multiple choice exam, it's going to be more like I'll describe a prokaryotic cell and then you determine is that description of prokaryotic cell or eukaryotic cell. Okay, so I'm not going to say, there will be no test question which says list all the things that make a prokaryotic cell different than a eukaryotic cell, right? So I know what that is, multiple choice. So here's some things that they all have in common. You need to know what they have in common. All cells have a plasma membrane, right? And the plasma membrane is an outer covering that separates the interior from its surrounding. In a sense, you might say it is what makes the cell a cell, right? Otherwise, it's just a bunch of molecules floating around in the liquid. But no, they're compartmentalized, right? All cells have something called cytoplasm, which is the jelly-like region where the other cellular components are found. So put these two bullet points together. All cells have a membrane, and inside that membrane is gunk, a cytoplasm, where all the other stuff can be found. All cells have DNA, which is, as we know from the previous chapter, our genetic material. There are different types. We'll talk about that later. All cells have ribosomes, and you're going to learn about that later in this semester too. So that one is actually pretty important. Ribosomes are the things that actually make the proteins. And again, we're going to talk about all these things later. This particular slide is like a preview. So any questions about this particular slide? All right. So examples. Here's some examples. Like I said, you should be able to know some examples of prokaryotic cells. Here we go. Basically, they're broken down into bacteria or archaea. Don't necessarily need to know. Don't need to know the others. So, but yeah, those are two examples. Not much to say about that. We will dig into them a little bit later, but as far as I'm concerned, that's all you need to know about that. Let's see. I'll take questions after this next slide. Let's show you an example of one, right? Here's the prokaryotic cells, right? Prokaryotic cells are simple, right? So again, that's a lot of comparing and contrasting. Our prokaryotic cell is different than eukaryotic, so I'm telling you now, prokaryotic cells are simple. As you might imagine, once we get to eukaryotic cells, it'll be the opposite, right? They're not simple, complex. Prokaryotes are single-celled organisms. Eukaryotic cells sometimes can be, but by definition, prokaryotic cells are single-celled. Prokaryotic cells lack nucleus, right? They don't have a nucleus, and they don't have any other membrane-bound organelles. And I haven't taught you what a membrane-bound organelle is yet, but for now, don't worry about it. What I'm saying is they don't have a nucleus, and they don't have some of the pieces that eukaryotes have, right? Eukaryotes have some organelles, right? The things that do work in their cells, and they have certain organelles that prokaryotic cells don't, because they don't have any organelles. They don't have any membrane-bound organelles. And then finally, for the DNA in prokaryotic cells, you can find it in the central part of the cell, in that darkened region called the nucleoid. They don't have a nucleus, they have something called a nucleoid. But once again, if I was sitting in your shoes, like, what? This is so much information, I might get a little overwhelmed. So I want to remind you for the exam, I'm not going to say describe a prokaryotic cell, right? I will provide this description. You need to say, oh yeah, that's prokaryotic, not eukaryotic. It's simple, not complex. It's single cell. It doesn't have a nucleus, right? All these things that make it a prokaryotic cell. Any questions about that before we contrast and look at a eukaryotic cell? All right. Eukaryotic cells, they do have organelles, right? Because I just got finished telling you prokaryotes don't have organelles. Eukaryotic cells do have organelles. And again, what do? They just have special jobs, and you're going to learn about those special jobs later. For now, I'll just know that they're pieces inside of the cell that have a job to do. And then finally, when we're talking about comparing and contrasting prokaryotic versus eukaryotic cells, there's a size difference. You can see prokaryotes are much smaller than eukaryotes. But I will put it next to this, through the numbers. Definitely don't need to know the size of a eukaryotic cell, which is 10 to 100 micrometers. You definitely don't need to know the size of a prokaryotic cells, which is 0.1 to 5 micrometers, right? You don't need to know that. You just need to know that eukaryotes are much larger than prokaryotes. That picture, I know it's hard to see on the slide, but it comes directly from your textbook, so you have access to it. Any questions about this slide? Okay. It's 10 minutes. You know what? Let's talk about the learning objectives, the introduction to this section, and then after that, when we come back on Friday, after having had the introduction, we can actually jump into it. That'll be the next word for attendance, is Friday. So again, this next section of the chapter is going to be all about eukaryotic cells. That's what we're going to talk about on Friday. The learning objectives when we come back on Friday are, when we're done with it, you should be able to describe the structure of a plant and animal cell. So sort of like I just talked about the difference between the similarities and the differences between a prokaryotic cell and a eukaryotic cell. Well, now, under this category of eukaryotic cells, we're going to do the same for plant and animal cells. You should recognize what they have in common, what makes them different than each other. At the end of it, you should be able to state the role of the plasma membrane. Talk about that when we get there. It's pretty simple. You should be able to summarize the functions of the major cell organelles. And again, I'm going to go light on that because we have whole chapters devoted to the important things that cells do. For example, as you're about to learn in this section, plant cells have things called chloroplasts. And that's where photosynthesis happens, right? That's how they make their food, so to speak. So even though we're going to touch on that in this chapter, we're going to have a whole chapter based on that later. So once again, for that reason, I'm going to go through these pretty quickly for this chapter. We'll talk about the important stuff later. Anyway, and then the final learning objective for this section of this chapter is you should be able to describe the cytoskeleton and the extracellular matrix. And once again, in my opinion, that is a lot less important. So that's the introduction. Yeah. So when we come back on Friday, we'll jump into eukaryotic cells. Does anybody have any questions about lab or anything? All right. So remember, if you're coming, you have to be there on time by 10. And if you're not coming, because you'll know, because I've left you a message saying you can't come, get with me. We got to figure out how the extra hoops you can jump through to get some of your points back. Any questions? All right, that's it. I'll see you either in office hours, in lab, or on Friday. Oh, if you came in late, make sure you email me and tell me you came in late, because right now I have you down as absent. Question.
me show you, how you know. Oh. I don't want to mess with you. Can you pull up the pre-lab? Wait. Right, oh yeah, that's the, yeah. So if you don't pull up, the last three. Okay, let's see. Scroll down a little bit, please. Go on, there's another comment. So controls, you see, there you go, see. You needed to do all these. I left a comment on such good saying you need to do it. I reminded you on September 9th. It's a learning curve, don't feel bad. Like I said, you just have to jump through some extra hoops to make up for it, but
It's,